Mr. President, Mr. Prime Minister, Dr. Leonor Beleza, members of the Champalimaud Fund and Foundation, Dr. Sommer and members of the scientific jury, and honored guests, as well as, in fact, the entire nation of Portugal. It is a great honor to be chosen together with Dr. Nathans as this year's Champalimaud awardees. I, too, salute the Champalimaud family and foundation for the decision to foster vision research and to recognize its importance. As Dr. Nathans has mentioned earlier, vision is one of our most precious senses, from which comes art, science, humanity, beauty, and practically all aspects of life. Most of the work in my laboratory for the past 28 years has to do with how vision begins in the eye. Vision begins when light is absorbed by the visual pigments, which Dr. Nathans has just described and has studied so elegantly. These pigments are present in light-sensing cells in the retina called rod and cone photoreceptors, with the rods giving rise to night vision and cones to daylight vision. Upon absorbing light, the visual pigments trigger a series of biochemical and biophysical reactions inside the photoreceptors. As a result, these cells send out signals to a network of neurons in the retina, which analyze these light signals to give us information about shape, form, color, motion, and other features of the visual signal. The signals are then sent to the brain for further analysis, allowing us to recognize instantly such complex visual images as the face of one's grandmother. When I entered the field, there were already many observations about the biochemical and biophysical steps that trigger visual signals in the rod and cone photoreceptors. But there were also uncertainties, puzzles, and discrepancies. I was fortunate enough to be in the right place at the right time and managed to turn these puzzling observations into a coherent and logical picture of what exactly happens after light absorption. In this way, the function of each of the many protein components in this first step of vision has become understood. As is often the case, basic scientific research begins as a pure pursuit of the logic and beauty of nature, but bonuses follow. In this case, the revelation of the initial visual process has led to the understanding of a large number of hereditary eye diseases affecting rod and cone photoreceptors and therefore vision, such as retinitis pigmentosa, Leber's congenital amaurosis, Oguchi disease, and stationary night blindness, to name just a few. Many of these diseases have turned out to result from genetic mutations in the many protein components taking part in the initial visual process and to lead to the degeneration or dysfunction of the rods and cones. With the cause of these diseases becoming understood, the hope is that many of them can eventually be treated. Indeed, attempts in this direction, including gene therapy, are already in progress. In a separate direction, my general interest in vision has led me to the study of a newly discovered photoreceptor system in the eye. Besides rods and cones, it turns out that other photon sensing cells exist in the retina. Unlike rods and cones, these other photosensing cells are designed not for recognizing visual images, but for the purposes such as telling us the time of day based on the cycle of light and darkness around us. Indeed, our ability to get over jet lag when traveling across time zones owes a lot to these cells. The existence of these unusual photoreceptors also explains the clinical puzzle that some patients who are unable to see objects because of the loss of rods and cones can still sense changes in the cycle of light and darkness around them and adjust the body clocks such as sleep and wakefulness accordingly. As Dr. Nathans has rightly pointed out, each scientific advance is built upon the success of others. My research is no exception. This award is first and foremost an encouragement and recognition of this important field of study and the many wonderful laboratories worldwide working on it. Likewise, my own work would not have succeeded without the great team members over the years, and this award belongs to them as well. Thank you very much.